information about the metabolites, about the biological implication of the metabolites, and also because the metabolites are actually an essential part of any living system. So that's why you can find it in many different areas I just briefly mentioned. So one particular area is in agriculture, plant research, or herbal medicine research, and this, in that case, and we typically try to actually uh, figure out what are the metabolites in a particular, for instance, plant sample in the seed or in the leaf, in the roots, and so on and so forth. So this is the steps you typically do to apply metabolomic technology. So essentially, you gather the samples, and then you put the samples into a certain solution, and then you use this kind of machinery. So this machinery actually breaks the ingredients of the materials, and then ionizing the ingredients, and then use some kind of essentially based on the electric field, put them into an electric field, a beam in the electric field, so that the ions can actually move at a different speed, and then become separated for the analysis of the ingredients. So that's the typical approaches. So of course, this technique can be applied not only for plant ingredients, but for any other type of bio samples. So these kind of things. So as I just mentioned, so we have a uh, some kind of broken up materials, pieces of materials from bio samples, and then we inject the broken up materials or the fragments of the materials into this channel with the and because the materials are ionized, so that means they are attached by certain charges, and that they can and then obviously they move along a electric field at a different speed because of the Coulomb force exerted on the ionized fragments. They differ depending on the charge and the mass. This kind of charge mass ratio. So because the difference in the charge mass ratio of the fragments, and then they become separate. Those moving at high speed, they move in the front, and then those slower in speed, they actually behind, lags behind, and then you can use some light beam to shine on the stream of the ionized fragments, and then that's going to put these uh, reflected lights, and then you can receive this light and display that as some kind of spectrum, like this one. So each peak corresponds to a particular fragment, and you can actually observe. So this is the process. Obviously, a peak correspond to some kind of fragment, and then and then you are going to have multiple peaks, each correspond to a particular fragment. Now you can feel that there is this task on how to figure out which combination of which peaks actually uh, makes up the original metabolites because in a typical biosample you can have many metabolites and then you're going to have many peaks of fragments. How to figure that out is really, you can see it is a daunting task, but this task is supposed to be handled by metabolomics. Okay, so this is a more detailed or close-up view of the channel that you can see the beam they become separated because of the difference in the speed, and then and also you can see that the, the the ability to generate the peaks also depends on the injection speed, energy of collision, and so on and so forth. So these are concerned with the physics because in the end you need to have it received and also so that you can have some sort of reflected light to record the spectrum. And also, you can break up the materials into smaller pieces, into fragments, uh, and at different levels. So the normal level gives you the normal mass spectroscopy. Mass spectroscopy is the process of the ionization of the fragments and then the force it to move along force them to move along the channel for the detection to detect the piece of mass. So essentially, this is the process, and then you have this secondary breakup to actually break them into smaller, more detailed fragments for more fine, uh, essentially. You have a fine green type of something like a type of jigsaw puzzles to actually uh, reassemble the original molecule, original metabolites. So this is the process. Okay, so okay, with this kind of process, suppose that we can somehow reassemble the metabolites from those MS peaks, mass spectroscopy peaks, we should be able to, and also we know that the metabolites, the type of metabolites, the level, actually the content of the metabolites, they are somehow linked to the biological conditions, or we can call it a phenotype. Phenotype could be, for instance, the aging phenotype and the environmentally affected phenotype, disease state, or the drug interfered phenotype, and so on and so forth, or the phenotype corresponding to different the nutrition state, whether it is a malnutrition or maybe it is over over supply of food and so on and so forth. These kind of things. So for human health, and we know that metabolomics are widely utilized for cancer research, for cancer diagnosis, and as well as for immunological studies, particularly because the immune system are actually in the blood system, right? And also because the blood samples are easier to collect, so if we can study the metabolomics of the blood samples, and if that the metabolic states are correlated to certain disease state or to certain conditions, certainly it can be utilized for the diagnosis of cancer and other disease. And also it could be applied for instance, forensic research for the study of the toxicity of the drug therapy, and also for, I think many of us are interested in traditional medicine, herbal medicine, and also we need to study why is that a combination of herbs and that they actually have a rather reduced toxicity instead of enhanced toxicity. So these kind of things could be studied by metabolomics. And also, the metabolomics could also be utilized for biomarker discovery. So certain metabolites uh, uh, they are maybe actually they are highly correlated to a certain disease state. So that means we don't have to measure all the metabolites. We can actually focus on specific metabolites. So this is essentially a targeted metabolomics. A targeted metabolomics is more focused, and obviously it can actually be give us the precise information and also at a lower cost. So obviously that is a very important thing. And another application is that, for instance, if we if we study for instance, the plant, the agricultural thing that plant actually grow with respect to time. We want to see the development of the plants, and then so that we can actually grow the plants more efficiently or grow the herbs to produce the more potent the actually bioactive ingredients, the more potent bioactive ingredients at a higher abundance, and that's what we want. So in that case, we may need to monitor uh, the dependence of the metabolite yields with respect to the developmental stage with the time. Of course, we could say the same thing on the disease development from the earlier stage of cancer to the later stage. So it is essentially the same thing. So this kind of called spatial temporal profiling of the metabolites. Obviously, we can actually apply the metabolomics approach to study this kind of to actually do this kind of tasks. Mhm. So in agriculture, not only we want to uh, actually find out or select those better yield products, the seeds of the better yield uh, uh, farm products or the plants, right? We also want to use biotechnology to, uh, for instance, we can actually apply genetic technology to actually modify the plant to make them uh, better in terms of yield, in terms of the quality of the nutrition. And then it is the same thing for the herbs. And particularly in recent years, people applied synthetic biology to actually transfer the genes from one species to another, so that the receiving species can have a more productive yield of something, a high quality of the products, and so on and so forth. Obviously, in these studies, the yield maybe the metabolites is an important part of the yield, and maybe we need to we can apply the metabolomics approach to monitor the change. And also, of course, not only because it concerns with the genetic manipulation, we also need to do other omics. So it is a multi-omics approach, including the metabolomics. Okay, in drug discovery, obviously for drug discovery, the measurement of metabolites of the patients is also very, very important. So, for instance, it concerns with uh, the resistance against the antimicrobial drugs. It concerns with the pharmacokinetics of the medicines the patients are taking, and so on. And also for some metabolic diseases, and certainly the metabolic products of the metabolic process or the metabolic targets need to be monitored. So these can all be handled by metabolomics. So here is an illustration of the application of metabolomics in drug discovery. Uh, so you can actually, for instance, you can actually monitor how the drugs are metabolized into metabolite products, and you can also monitor the change of the other metabolites, for instance, in an animal model to see, for instance, in the blood. For herbal medicine, traditional medicine, people typically people need to measure because a traditional medicinal recipe typically are composed of multiple herbs. Each herb are with multiple plant ingredients, right? So those multiple ingredients and the best way to monitor those ingredients inside for instance, an animal or human body is the measurement of the metabolites in the bloodstream, because uh, the ingredients must be absorbed into the patient's body and into the bloodstream before those ingredients can be distributed to the targeted tissue for the kind of symptoms you want the medicine, be it traditional or modern medicine, or you want it to reach the target site, but certainly it have those ingredients have to go through the bloodstream, so you can apply the metabolomic study to study to monitor those ingredients. The first thing is that whether those ingredients they can enter into the bloodstream, whether or not they are modified either by microbes in gut microbiota, or actually modified maybe by some enzymes in the liver or in the other parts of the human body. And also you want to measure the pharmacodynamics of those multiple ingredients. Obviously, the best approach is this metabolomics approach so that can actually monitor the many ingredients. Okay, because of the importance of the metabolomics technology, so actually there are this kind of they already developed protocols. So you can just follow these protocols to actually complete uh, the metabolomics study of the whatever things you want to study. Before we get into the AI part, and I want to further highlight that in metabolomics, there are so-called targeted and untargeted. A targeted, actually, I already briefly mentioned a targeted metabolomics actually simply focus on a subset, a selected set of metabolites. That means you know what metabolites you want to zoom in, you want to focus. And then, for instance, if it is so, this is an illustration of the multiple metabolite pathways in a living system. And if this is the part you want to focus, or if this is part you want to focus, and then you can actually zoom in on the metabolic products of these parts. And then because you know those kind of things, it's easier to reassemble the fragments. I will revisit this thing. Okay, actually, I will mention it just. How come I cannot move this one? It's very inconvenient. Okay. Okay, so I will mention it slightly later on. Okay. So in contrast for a untargeted metabolomics, essentially we do not have uh, a particular or a set of metabolites in mind. We don't know. For instance, we have a new herb we want to study. We don't even know what kind of ingredients the herbs have. So actually, it is the task of this metabolomics. You are supposed to tell us instead of we tell you which to focus. So in this case, this task is on target metabolomics. So that means the somehow the metabolomics methodology has to figure out, be it an AI or traditional bioinformatics method, no matter which method, that method has to figure out all those MS peaks and then reassemble them into many metabolites. But this one is really the task is really daunting. Okay, we look at the search space. Suppose we have n number of peaks. We know that so n number of peaks. So this is not a peak pair. N number of peaks. And if we minimally assemble two peaks into a metabolite, at least that means we have to assemble peak pairs, right? So suppose each peak to have essentially n possible n1 possible possible fragments it corresponds to, and then you need to have n1 times n2 times n possible peak pairs because we don't know which peak and with which peak to form the pairs. And also, so this is the theoretical peak pair, the fragments we can assemble out of n number of peaks. And on the other hand, we have the library fragments, which actually we need to use our theoretical pair to compare with the real chemical structure, right? So supposedly our library has m number of, sorry, these fragments, and then total number of possibilities m times m1 times m2. So it depends on what is these numbers. So if we do with targeted one, certainly we have a smaller number of uh, fragments. So we have smaller number of arms here. Uh, so certainly it drastically reduces the task. On the other hand, if we do with untargeted one, sorry, we have to consider all possible chemical structures libraries. Okay, later I will visit this. What are the peaks pairs? What are the fragment pairs? So right now I just give you some signs that it is actually you can see it is a exponential. It corresponds to an exponential relationship of these kind of things. And also, technologically, if you use untargeted metabolomics, there are multiple techniques, multiple steps you have to use. For instance, you have to use the NMR and a few other techniques. But for targeted one, you don't have to be too complicated because you only focus on a smaller number of fragments, and those fragments can only give you a limited, highly limited number of theoretical peaks because you can assemble theoretical peaks. And so you know, so even the number of peaks are actually, you can zoom in, you can actually, it's actually constrained. We go back to this one. Okay. So even this part is reduced because this part concerned with the theoretical fragments, this part with the real fragments. Okay. Uh, so far we talk about the untargeted metabolomics, and so you can use that and for further analysis. And you also have this targeted metabolomics approaches. So for targeted approach, remember that I mentioned that we can actually do time dependent. So you see this clock here, time dependent analysis. So for time dependent analysis, we can afford to do targeted, but we cannot afford to do untargeted. And so that's why you can have this complex system of a combination of untargeted and targeted metabolomics, and so that you can not only you can trace the knowns and unknowns, but you can also study the time evolution of the metabolomics states, this kind of these kind of things. And also, you can actually link the metabolites with, for instance, with the pathway analysis, and also you can actually apply the statistical data analysis to generate the pattern. Remember that we have t-SNE, those kind of things. UMAP, actually, you can actually do all sorts of analysis. Okay, so far, so much so about the metabolomics technology. Now we want to jump into the main topic of our lecture series, right? The AI. So first we look at the machine learning part of the metabolomics. Okay, remember that we talk about the metabolomics works by breaking up the metabolites into smaller pieces. And then so that's this question: how, in where and how much the metabolites are fragmented. Obviously, it has this kind of fragmentation in layers because you need to break up a certain bond. For instance, if you break up it here, and then you have this part almost like this, and then if you break this one, and then you see this one is broken, you have this kind of structures. So that means there's not a single solution of the sets of fragments. You can actually have many fragments. Even a particular part may actually. So if you consider as a subframe, even for this subframe, you can see it can have many different fragments. So that's why it actually forms a so-called fragment tree. You can see there are multiple choices, multiple ways to break up the metabolites into this kind of tree structure. So you need a fragment tree representation to represent the all possible fragments. Obviously, some fragments, of course, you can theoretically break things up, but the question is whether those kind of fragments can really be realized, really be produced by the metabological machinery. Uh, so that's why you need a, a system to train the fragment tree to make sure the fragment tree you generate it's really can really occur, can really appear in reality. So these are the examples of the peaks of the fragments. Okay, so maybe let me mention a little bit about the peaks here. So given a peak, so each peak actually you can see the x-axis is the ratio of the mass and the charge. So if you fix the charge, so essentially it corresponds to the value of the mass. Given a mass, we can figure out we can figure out what are the possible fragments correspond to this particular mass because if you fix the value of the mass, the number of atom types are limited. Okay, I just stop here. Later on, I will get back at this point. How do we? So essentially, it is if you remember in chemistry we use the molecular formula. If you use the molecular formula, it's easier to understand why, given a fixed mass, the number of possible fragments are fixed. We will come back to this again. But now we further go on this kind of AI machine learning method for uh, metabolomics analysis. So metabolomics analysis, the task here is to start with a uh, mass spectroscopy peaks, and then we need to figure out, we need to assemble those peaks into the fragments, into the molecules for us. So of course we have this question, which peak combine which peak to form molecule one, which peak combine with which to form molecule two, and so on and so forth. So starting from the peak, and if we have a trained system, we can easily generate this kind of MS tree. If you don't have a training system, you can also generate this tree manually. So now I come back to this. Remember that I mentioned that each peak corresponds to a particular mass. We actually assume the charge is fixed for all. So actually it is the variation of the mass along the x-axis, right? So so each fragment must be a composition of different items. For instance, for water, we are familiar with H2O, right? Two hydrogens, one oxygen, right? Water, H2O. We know the mass of H. Obviously, we know the mass of H2, we know the mass of oxygen. Obviously, we know the mass of O. So water H2O is simply the mass 2 H plus the mass of 1 O, right? So that's why you can actually easily write down all possible molecular formulas. So H2O is the molecular formula for water. And then provided that the sum of the mass is equal to the mass of this particular peak. So I think you can understand that there are only limited molecular formula, for instance, like H2O. So you can think of, you can determine the mass of H2O, right? And then you can ask the question, what the other molecules gives the exactly same mass as the H2O. You can figure out whether uh, obviously if you can design a molecule of that contains only hydrogen, obviously you can add 16 hydrogen to replace the oxygen, and you can get a equivalent get another molecule of equivalent mass. Of course, so you can't use all hydrogen to form a molecule. So that's another issue here. So anyway, you can think that we can generate this kind of fragment tree, and each is marked by a particular molecular formula of a particular molecular mass. Obviously, the lower level of the fragments, they must have a mass smaller than the top one, the root one. This is the root, and then you have the branching out. So the mass is further and reduced. Anyway, you have this fragment tree, and then you can develop a feature. So in this case, the authors developed these kernel features and then send it to SVM to train a machine learning method SVM to actually predict the. So essentially, you can design a molecular fingerprint. Remember that molecular fingerprint is essentially 1010. So each position corresponds to a particular type of fragment. If the this kernel feature, this molecular tree, fragment tree feature has a particular fragment. This one actually corresponds to one, right? And then with this 10101, it actually gives you the molecular fingerprint of the metabolites. So assemble the metabolites. So this is how a machine learning method works. You input the features of MS peaks and trees, and then you output a molecular fingerprint, which actually corresponds to the metabolites of the combination of these traits, and so on and so forth. So this is how it works. It works. Uh, you can have so remember that this kind of AI prediction, AI generated molecular fingerprint is a theoretical fingerprint because it is derived from the fragment. You actually assume a fragment has this kind of mass, right? So this mass is theoretical. But there's this question. Suppose that, let it put it this way. Suppose that we can assemble the same so sorry, we can assemble the similar fragments, fragment A, similar metabolite A using these two peaks plus this one. We can also assemble another almost the same molecular, so this one with a lower peak here, lower mass peak here. And then we can assemble metabolite B with a slight difference using these two without a smaller peak here. So you have two solutions here, but which solution is valid? Obviously, it is up to the chemistry. That means that theoretically you can, so that means theoretically you can add another fragment onto these two fragments. So let it put this way. So each peak corresponds to some kind of fragment. Uh, but you need to ask the question whether chemically it can really happen. If not, then that means that fragment actually belong to somebody else. Maybe that fragment should be given to these two instead of these two, right? So how do we know that? So that's why we need a chemical library, chemical database, like PubChem, for instance. And then, so those chemical database also gives us many, many molecular fingerprints. So we need to ask the question whether the AI generated molecular fingerprints correspond to something in the library. If yes, we are very happy because we know it is confirmed that that chemical molecule can happen, can actually exist in reality, be it uh, human synthesized or it is produced naturally. So obviously, because we do metabolomic study, typically we assume the metabolites are from nature, but we cannot rule out some metabolites are the man-made chemicals like drugs taken up or somehow uh, distributed into the environment. So those man-made chemicals. So we cannot rule that out. Okay, so that's the first machine learning method. Now we look at the second one called cosmic. You can see that this part is essentially the same one as I just mentioned. That method is called CSI finger ID. Essentially, it try to generate a molecular fingerprint out of the mass spectroscopy data, essentially. And then this cosmic machine learning method goes one step further to actually further try to further score, has a more sophisticated AI system to score the quality of the molecules generated by the molecular fingerprint. So that's one thing. The other thing is that the molecular fingerprints doesn't really give you the molecular structures. Through a sophisticated machine learning method, in comparison with a chemical database, you can, if there's a match, good match of the molecular fingerprint, actually you can obtain the molecular structure based on the molecular structure in the chemical database because they are matched. So that's why, so this machine learning method actually not only output the molecular fingerprints, but also give you the match the molecular structure. So essentially, it acts as some kind of prediction or some kind of generation of the molecular structure for you. But provided, so it's not really generative AI because this structure must exist in a chemical database. So this is the second machine learning method. The third machine learning method, so you can see that that's this machine learning method. It tries to actually predict the so essentially it tries to focus on the substructures of the metabolites and then give you a rank of those substructures so that because we know that there could be uh, for instance, the same metabolites could be assembled by different substructures, right? And then there's this question, and also a slight variation of the substrate composition can actually give you a different derivatives, even though the framework uh, is unchanged, is the same, but the derivatives can be different. So there's still this question, how you can better rank the system. So the previous paper actually uh, uh, directly rank each assembled molecule, and this one tries to rank the fragments as well, not no, just rank the uh, the complete thing. Okay. So the next method is called body method. So this method is even clearer, right? So you have MS peaks. So each peaks you see this molecular formula, right? So each peak has a mass. You can easily figure out the molecular formulas. But so each peak could have multiple molecular formulas correspond to the same peak or the same composition of the peaks. So the question is, which one goes to which? How many peaks should be the real one? So you don't know the which fragment because each theoretical, so each molecular formula or called the chemical formula, each formula can actually correspond, theoretically can be associated with some combination of peaks. So all these are theoretically valid, right? So which we actually mentioned in the introduction of the previous machine learning models. So in this machine learning model, it tries to have some kind of ranking system to actually rank the molecular formulas for subsequent analysis. Okay, so this ranking, so actually you have the molecular formulas ranked against the different peaks. And then this, remember that the previous method tried to have something to score the fragments. So this method actually is similar, try to score uh, from the perspectives of the peaks, because each peak corresponds to a theoretical fragment. So you have this kind of score for each theoretical fragments of each peak, and then you can combine the collective score to get a better uh, final outcome. So this is the approach. So you can see this is the summary, the peaks theoretical formulas, and then it tries to evaluate the actually the match from the perspective of the theoretical peaks. So how do you do the ranking? Remember that all the ranking has to do with respect to the real chemicals in the chemical database. So you still need to have some chemical base for database for uh, this kind of ranking scheme. Because of the time, we are not going to, and also it's very complicated. Not only because of the time, actually, uh, the the author didn't disclose too much about how they uh, actually figure out the ranking. They just give you a very rough description about the computational procedures. Okay. So far we introduced several machine learning methods. Those methods are actually quite popular, but uh, the usefulness of those methods are actually up to uh, different opinions. I discussed the usefulness of the AI, particularly this, because these machine learning methods, they are more popular because the deep learning methods are still in the development. So I discussed this with some elite scientists in the sequencing established a sequencing company in China because they actually do sequencing, metabolomics, measurement, and analysis for customers every day in a daily basis. So they said that they tried all sorts of AI methods, and so their conclusion is that it's not really uh, up to the stage, up to the level that it is a really reliable method for them to actually to use as a daily on the daily basis. Certainly, it's not up to that level yet. So this is but we can't discount that. Remember that AI, good AI, needs lots of data. We cannot say we have a large enough metabolomic data to train good AI. Perhaps not. So that's why uh, we we expect as the data, so big data, as the data becomes more available, more and more available, we could have an AI that actually be used as a on a daily basis. Of course, we wish that AI to be deep learning because deep learning, as long as we have big enough data, deep learning definitely is a better approach. We want to see how so far how deep learning are utilized in this case. You see, this is a series 4. This is a web server. They actually added the deep learning. Of course, they still have machine learning. You see this familiar part, right? So you have this MS3 peaks, and then you see this machine learning part to generate the molecular fingerprint for you. And also you can have this, remember that the kernel features for just like this one, right? You can actually send the same kernel features into CNN in the deep learning model to generate molecular fingerprints. So you have two sets of molecular fingerprints, and then you can compare the molecular fingerprints with a chemical database to find a match, to know that the theoretical molecules can really appear in reality, and also you can happily generate the chemical structure based on the structure of the database, match the chemical. So you do the same thing for deep learning and also for machine learning, and then you can combine the wisdom of two to actually, for instance, if both of them are consistent, you have a higher confidence the the generated molecule is really the metabolite you are looking for. If they disagree, and so it is up to the certainly you need a training set, so you need to assign the different weights, whether the higher weight should go to CN or go to SVM. It depends on the training set. So it is a combination of both to make a decision about the final outcome. Okay. So now we look at the second deep learning method, Canopus. So this deep learning method actually so you see they still have this SVM part to generate the molecular fingerprints, and then the molecular fingerprints, and you can also get the molecular. Of course, you can get the molecular fingerprints from the chemical databases, and you use those molecular fingerprints and also those sorry the molecular fingerprints of the chemicals, but those chemicals in the database, they actually have some kind of functionality information, like, for instance, inhibitors of EGFR or anti-cancer agents or whatsoever. So essentially, you can input the molecular fingerprints of the chemical database and output the labels of the molecular fingerprints. For instance, in this one, the molecular fingerprints are associated with lipids or lipid-like type of things. These are the examples. So you can see that machine learning take care of the generation of molecular fingerprints, and deep learning take care of the type. So what is the functionality of the molecular fingerprints? So this is the approach. So once you have a deep learning model, once the machine learning model generates a predicted molecular fingerprint, you can input that molecular fingerprint into the deep learning model and to generate a label to tell you what kind of molecule it is. So this is the uh, application of a deep learning model for uh, functionality labeling of the machine learning predicted molecular fingerprints. Okay, so this is a more detailed architecture for generating the labels, the molecular functionality, or the chemical class, or you can call the chemical class of the molecular fingerprints. And of course, depending on how many chemical class, how many functionality you are interested in, you could have many classes here. So it is a multitask deep learning model to generate the kind of information, essentially to label the output molecular fingerprints for the users. And because of the ability to generate the chemical class label, for instance, if we label a chemical class as EGFR inhibitor, so this method can be utilized for molecular design. Suppose we have a molecular the framework, and we can add or subtract different substructures here, right? Different substructures, and then we can uh, use the deep learning part to see whether they belong to the class of EGFR inhibitor. If some substructure belong to, we know that we actually generated new derivatives that actually have the kind of activity we want. So this is a example of the applications. So uh, so the generative AI becomes useful because you can use the AI to generate the molecular the different so the same framework with different molecular substructures, and then you can check the activity using this method. Okay, so the next AI actually adopts new architecture, transformer architecture. So the starting point is again the mass spectroscopy peaks. Remember that each peak can be associated with a chemical formula, molecular formula here, right? And then because there are many peaks, you have many formulas, right? Of course, you can use this MS-tree uh, representation and then send it to a SVM to produce molecular fingerprints, right? So this is a machine learning approach. So we already see that quite a number of AI methods actually use this one as a part of their more complicated, more sophisticated system. Here, Even though they have transformer, they still have this component, important component here. Okay, so what is the task of a transformer? So essentially, remember that we have all these possible formulas for these peaks. So we can actually consider each peak as a token. Each peak is associated with a formula. Each formula, remember that they are some kind of code, right? So this is what we are familiar with. So a transformer is happy to receive the code, and then you can convert the code, the codes into some kind of coding system, one hot encoding, and then into some kind of word embedding. Remember those things. And then certainly you can use this transformer, simply use the uh, each peak as a token, and then the molecular formula, the coding of molecular formula as the input, and then you output the molecular fingerprint. So this is one application of the transformer to generate the molecular fingerprints. And then so again, you have two molecular fingerprints, and then you can actually use this consensus modeling weighted constellation approach to select the best molecular fingerprint. Once you have the molecular fingerprint, these are the applications you can use the molecular fingerprint. Remember that to actually classify the chemicals. For instance, I mentioned the EGFR inhibitor, those kind of things. You have these kind of applications. You can also to do this kind of genome matching, those kind of things. Actually, we may actually we may study some of aspects of genome matching in our metagenomic study, but not too much. Just okay. So that's the some topic. Actually, that's something else. We may actually mention a little bit in that, not too many. Okay. So the next method is called MIST. So in this method, again, you see the transformer are used. So the transformer, they are used here. You see the transformers, right? They are used here because remember that we have the peaks, and we see that we can use the peaks to encode them either as some kind of chemical formula or as some kind of MS tree. Remember that, right? So people thought that as long as we have this coding thing, it's natural to send it to a transformer. But not only that, because we also have some other representation, like the theoretical fragments and so on and so forth. We could also jointly send those part of the information into the transformer. So this transformer not only accepts the coded peaks of the formula, but also some other features into this transformer. Once the transformer receives that, it can actually output different things. Uh, for instance, we could remember that the transformers are really initially developed as a pre-training model. That means we input something, we reproduce something, and then we mask some part. We produce that part without masking, right? So you see, this is exactly the same thing. So we have a pre-trained transformer, we input the coding, and we output the coding. Once this pre-trained model is developed, we can use the features for the subsequent tasks, like the generation of the molecular fingerprint, or the generation of something else, like the activity and so on and so forth. So with this kind of pre-trained models, we have this kind of uh, output, and also remember that we have remember that we for each peak we could have multiple chemical formulas corresponding to the same peak, right? So you have multiple ones. Uh, so that's why you could have a minimum of one solution, but you could have multiple solutions. So those multiple solutions, even though they may not be the so-called the in existence in reality, but we never know whether they may exist in the future. So that's why you can put them into a library, either as some kind of decoy, meaning that they may be fake ones instead of real ones, but you never know. So that's why you put them into a decoy library along with the real chemicals in the PubChem. So they may still find some usefulness in the future. So that's the so anyway, we really don't know whether a particular combination can really be synthesizable. Even though we human can't synthesize, it doesn't mean a microbe cannot synthesize that. So actually, we have many natural products. So we don't even we have not been successful in synthesizing that. Natural product synthesis is really a very difficult task, and many medicinal chemists they actually devote themselves to do this natural product synthesis. We never know. So that's why we it's not actually it's a good thing that we keep them instead of throw them away. Okay. Okay, the next deep learning method, again, it utilizes a transformer kind of thing. It uses transformer uh, because it wants to actually to move this, actually block my way. Uh, so it actually wants to be able to directly generate the. Remember that the previous ones they actually generated the molecular fingerprint, right? And then you need to use a chemical database to figure out the chemicals. So this one try to uh, get rid of that limitation. Why should we need a chemical structure database in order to generate a chemical structure? Why not we directly generate a chemical structure? So in this study, they actually use a transformer to output not a not only a molecular fingerprint, but rather a smiles structure, smiles coding. So that's the type of output smiles. So this is a nice try. How successful it is, it's up to the really up to different people's opinion. So it's very hard to obviously it's certainly not as good as generating a molecular fingerprint. Definitely, this one is a harder task to do. But anyway, it is a we need to go along this direction because we want the AI to be able to generate the structure directly from MSPs instead of using some chemical database, structure database. So this is a more detailed layout of the.
Excuse me, Professor.
Yes.
We have a time is almost we have a short, so please wind up in a 5 minute because the 1 hour is almost done.
Okay, sure. Actually, just one or two slides. It will be
Okay, okay,
finished.
okay. Take your time. Okay, thank you. Thank
Okay,
you.
okay. Okay, so this is the detailed architecture. So this is the kind of molecule. You see this smiles code. It actually generated for you. So this is the thing. Okay, that's all for today's lecture. Now is the question time. So I will send the PPT. I already sent it. So I sorry, I sent the PPT late today because I there was a meeting in another location of Ningbo. I just came back from the meeting, so I didn't manage to send the PPT in the morning. So anyway, I just sent it to you, and also I will send the other lecture notes tonight. Okay. Mm.
Okay, thank you, Professor. So it's question answer time. Let me
Mm.
ask the question answers in a physical participants. Do you have any question? Okay, please come to the stage. Please first introduce yourself to Professor and then ask the question.
Hello, Professor. I'm
Oh, yeah.
Narendra from NCP Islamabad. I have a couple of queries to ask regarding the metabolomics for the drug discovery.
Mm.
How does metabolomics help in distinguishing drug metabolism, or you can say the biotransformation, from the drug mode of action during the drug discovery?
It's a good question. As I mentioned that a drug can be metabolized into different derivatives, and then certainly you can use a metabolomics study to actually to find out what are the metabolites. There's this question of whether you know the metabolic products beforehand. If yes, you can use targeted metabolomics so that you don't have to actually search too much metabolic space, right? So if you don't know, and then you have to, of course, if you focus on particular drug, certainly you know the metabolites, the chemical space or metabolic space is somehow limited, so that you may design the range of the mass, molecular mass you want to search. So that way that can also save time. So actually, in order for a sequencing company to do this, they need to design a library beforehand. So if you or if you are capable of designing a metabolomical platform yourself, and you need to design this library so that you can do this focus analysis. Without this focused study, then you have to do on target. I can tell you the on targeted metabolomics, they are their ability to identifying metabolites, particularly the metabolites of largest molecular size, is really limited. The capability is quite limited.
Okay, thank you so much. Okay, I have another question. As you have mentioned our article about the systematic classification, I have a question regarding this. If you can answer briefly or to the point for this, for the systematic classification of unknown metabolites, what type of you can metabolomics we can target toward, targeted or untargeted?
As I mentioned that, actually in machine learning method, they also have this fuzzy machine learning method, meaning that you can make the system a little bit more flexible within certain range. Then certainly it can both search more space for you, either chemical or biological space. and also without drastically increasing the computational load for you. But unfortunately, at least for many sequencing companies, sequencing groups, they can't really, or omics groups, they can't really customarily do that for you. So maybe you have to do it yourself based on your intuition, based on your knowledge. Uh, for instance, certainly a clue is the restriction of the molecular mass. If you do that, certainly the number of peaks you have to deal with is really quite limited. But if you don't know, if for instance, yeah, if your drug molecule can really break into very small piece plus bigger piece and so on and so forth, then and then the spectral space is actually is quite you have to quite extensive. You have to include many different peaks because you never know which one is which. And then it becomes a difficult thing. So that's why most people tend to focus on known metabolites, known derivatives. Things are easier that way. Okay.
Okay, thank you so much, Professor. In any,
Okay.
anybody have a question? Do you have? Please come to the stage. Please introduce yourself, Professor.
Hello, Professor. I am Fazal Wahid, and I am working as a research assistant at the Qaid-e-Azam University. I have a question about how do you see the transformer-based architecture, which is performing in the metabolomics compared to the CNN, which is more established?
It depends on the training set and transformer. Remember that if you work on the peaks theoretical chemical formula, and you automatically get those coding, right? The chemical formula coding. And then, so it is a natural thing for a transformer. So that's why people thought, why not just use transformer? If you use CNN, CNN is good at capturing, because it doesn't deal with some kind of language thing. It deals with some kind of pixel thing, right? So, for instance, if you assume uh, if you can somehow featureize the input as some kind of vectors, and then you can apply the CNN. But CNN actually use a filter to grab the local features. So that means if your peaks, because local feature presumably still capture some combination of peaks, right? So if those nearby peaks, they are really nearby or they really belong to the same metabolites, then sin definitely is the best approach. However, if in most cases it is a smaller plus a bigger fragments, you can imagine that they are far apart. They can't be grabbed by a single filter, right? But a transformer may be able to deal with that because no matter how far apart, as long as the attention mechanism, attention algorithm is able to link them together, and then you know that the AI actually can grab those together. So it's really hard to say, but people thought that the transformer should be better in this case. But there's a downside with the transformer because transformer architecture requires more training data, requires more computing resource, which you may or may not have that. So if you put that into consideration, sometimes you may find that even if scene only captures some local feature, but you can have two local filters later on. They as long you go into deeper, right? Deeper layers, later on those filters may actually eventually get together, and then you can still actually link them together. Okay, so as far as mass spectroscopic data are concerned, many data they are actually unlabeled. So that's why typically if you use transformer, you can do this pre-training type of things. But for CNN, typically you need to have labels, but CNN actually doesn't need too many samples with labels. So you can see that it's really case by case. We can't say one method is superior than the other. It depends on the situation. Okay.
Okay, thank you so much, Professor. That is all for today. The
Okay.
lecture was so great, and we'll see you on Monday.
On
Have a
Monday.
nice weekend. Bye bye.
Okay, bye bye. Have a nice weekend. Bye bye.